why we prepare a bacterial smear is to stain them so that we can see them and we can identify them. It's a regular diagnostic process. So one of the very important aspect of bacterial demonstration is bacterial stains and staining technique. As you know, where from this concept is will appear is that issue, I need to admit them. Okay, what's happening? Okay. Under phase contrast microscopy, but like that brightscopy, where the microscopic field will be brightly illuminating. So in that case, all the light will pass through the bacteria. As a result, you can see the bacteria under the microscope. So until and unless you put some color to the bacteria, you cannot visualize them. And from there, this concept of stains and staining technique has come. And after putting some color to the bacteria, it will be so contrasting that in a white background, in a bright field microscopes, you can see some colored object, and that you can very well appreciate its shape, size, structure, organization, arrangement. So that help us in identification of the organism. Not only that, in the development stages of bacterial stains and staining, some staining techniques has developed what we now call as the differential staining. This differential staining technique not only will able to identify and see the organism, but at the same time it can differentiate the bacteria on the basis of their certain cell wall characteristics. What you know about this ground staining techniques, which can differentiate broadly all the bacteria into gram positive and gram negative. Similarly, we had some differential staining to demonstrate a special kind of bacteria causing tuberculosis in man and animal. It's the Mycobacterium species, which usually cannot be demonstrated by ground staining than this alternative acid fast staining. So these are the two classical examples of differential staining. Of course, we have many more other differential staining, but most commonly we use this two differential staining. So in this particular class, we are going to learn about this organism, the stains and staining. Okay, I made this mistake. So before going to the techniques, let us first understand about what are these stains and how we can impart the color. It's not that all the type of color available with us we can apply to use to stain a bacteria. Okay, we apply some paint in the wall, we paint in air canvases. So do you think this all are we can apply it for bacteria? Simply no. Like the stains are certain special dye compound, and this stain and dye compound, first of all, they must possess some very small particle size, so that they can diffuse across the membrane and can stain the bacteria, what we call as the positive staining. So this stain and the dye compounds, they must possess two different group. One is called as the chromophore, other is oxochrome. Chromophore imparts color to the bacteria, whereas the oxochrome group help in electrolytic dissociation. So a dye compound must possess both chromophore and oxochrome groups so that they can react with the cellular components and can impart color to the organism. It should not be that if you just wash it in the tap water, all the dye compound will wash off. So a dye compound containing oxochrome group will not easily decolorize when you wash the bacterial smear in the tap water after putting the color. So maybe some ordinary dye compound which doesn't have this oxochrome group may wash off when you just wash the bacterial smear in the running tap water. So likewise, we must need the chromophore and oxochrome group to be call it as a good dye compound for pertaining color to the bacteria. So if you go through the different types of dye compound available to us, basically they forms under this triphenyl methane dyes, oxazine dye, thiazine dye. But what we feel that it is difficult to remember this dye compound on the basis of its chemical composition, because the chemically they are much more complex in nature, and most of the time it is difficult to even remember the chemical nature of this topical compound name of this dye compound. So as a result, some other easier techniques has been developed as an acidic dye, basic dye, or neutral dye. Likewise, all the dye compound that we use it for pertaining color to the bacteria can be grouped. So when we say it's an acidic dye, and that indicates that it is having a negative charge on the dye ion, as the oxochrome group is having negative stain, negative ion. As a result, this acidic dye can put color to the negatively charged cellular component. Similarly, the basic and which is an positive charge compound can stain the nuclear component or the negatively charged component, vice versa, like that way. Whereas there are some dyes which are neutral dyes, like eosinate methyl blue. This kind of dyes are available, which can pertain color to both negatively charged and positively charged component of the bacterial cell wall, as well as the interior nucleic acid. So depending on our use, we can use any one of this component for its staining purpose. Okay, so again, coming to very simple, simplified form. Anyone else join again? Yeah. Oh, excuse me. There is some problems in setting this computer. I could not enter all of you before the class. That's why. As in when you're, yeah, again coming, I need to enter it. Okay. Could you see the slide?
Yes, sir.
Yes, sir.
Okay. So to understand a particular methylene blue chloride, it is an neutral dye compound. Methylene blue chloride, where the methylene blue is positively charged and chloride is the negatively charged ion. And this is a common dye that we use to impart color to the bacteria. So bacterial cell also it is a combination. Say bacteria, they are negatively charged with some sodium salts are present iodine compound. When you stain it with methylene blue chloride, this bacteria will combine with the methylene blue, negatively charged component of the bacterial cell will combine with methylene blue. Okay, only the negatively charged particles of the bacterial cells will combine with methylene blue, and methylene blue will impart color to that. And this chloride will again form a salt with sodium ion and electrolytic dissociations will occur. As a result, the bacteria will take the color properly, and if you, after putting the color, if you wash this bacterial smear in the running tap water, they will not wash off. So they will put the color will retain in the bacteria, and you can visualize them. This is a very general type of concept how this stain compound works. Okay, so we will be focusing much on the staining techniques and how best we can apply these staining methods for understanding the bacteria, to identify them in the diagnostic process. So as such, what you need to remember is that these are the different staining techniques that very frequently we use in the laboratory for demonstration of bacteria. So first kind of staining is called as the negative stains, and rest three are called as the positive stain. Example, the rest tree. Example are examples of positive stain. What the negative and the positive stain is, that in the positive stain, the bacteria will actively take the color of the dye compound. Suppose if you put some methylene blue color to the bacteria, all the bacteria will take methylene blue stain, and you can see the bacteria as blue color. That's what we call as an positive stain or active stain. Whereas there is a technique in demonstration of bacteria under the microscope, what we call as the negative staining. In the negative staining, the bacteria, in fact, will not take any color. Will not take any color. The dye compounds are so bigger in size that they cannot enter through this wall of the bacteria, and the bacteria will not take any color. Rather, the background will take a color, black color or dark color, and bacteria will be refractile. This is also a type of staining, what we call as the negative staining. More particularly, the subsequent slide will see this staining technique is used to demonstrate the capsules of bacteria. By now, you came to know about the capsules of bacteria. Some amorphous gelatinous material get deposited surrounding the bacteria, and this is an important features of the bacteria. Some of the bacterial capsules are too toxic for our body, and they can cause massive damage to our body cells because of the presence of these capsules. So in theory, you have learned about it. So how to identify this bacteria is having a capsule or doesn't have a capsule? We can go for negative staining to demonstrate it. So for that purpose, use this negative staining technique. Remember here, the bacteria in fact doesn't take any color. Rather, the background will take the color and the bacteria will be refracted. This concept is somehow similar to that of the dark field microscopy or phase contrast microscopy, and that we use it to visualize live bacteria. In the previous class, we have seen the motile bacteria and this phase contrast microscopy. So if you don't have this dark field and the phase contrast microscopes, then simply by performing negative staining, we can demonstrate the capsules of bacteria. Okay, so it's performed only for demonstrating the capsules of the bacteria. Whereas the other tests are other staining techniques are example of active staining, where the bacteria will take the color and we can see them under the microscope. Their shape and size can be differentiated, and the whole background is white, exactly just like this slides that you have seen. Everything is white, and the colored things you can see it as a bacteria. So simple staining is the second example of staining technique. Simple staining basically we perform if you just rapidly want to see whether the shape of the bacteria. So we just pick up one dye compound, put it on the bacteria. The bacteria will take the color, and then we can see the bacteria under the microscope and can find out whether it is a coccus, it's a bacillus, it's arrangement, streptobacilli, streptococcus, or staphylococcus, or it's a spirillum, etc., etc. We can just understand the shape of the bacteria. That's what we call simple staining. But simple staining doesn't give us much information as it can be provided by differential staining and the special staining. So simple staining, sometimes we perform rapid identification of the shape and arrangement of the bacteria, where we use a single dye compound. That's why it is called as the simple staining. Only one dye compound we use it. Then comes the differential staining. This is the most widely used staining technique in microbiology laboratory. This staining technique can identify the bacteria, their shape, arrangement. Not only that, on the basis of the cellular composition, we can identify the bacteria, whether it is a gram-positive or gram-negative. We'll come to this point in the subsequent slide. And lastly, it's come the special staining. Some staining techniques we adopt to demonstrate some metachromatic granules present or volutin granules present on the cytoplasm of certain kind of bacteria, which is diagnostically important. Not only that, you have learned about the endospore forming bacteria. We can just demonstrate by this kind of staining technique. The location of endospores are also important in identification of bacteria. So this staining techniques, we are going to discuss it. Recently, Brookes, he contacted me and asking me about the slides. I will provide you all the slides, and no need to take any screenshot and keep it for your use. I will provide this today itself in our group. Okay. No. Okay. Now coming to this negative staining, the example you need to remember, stain called as a nigrosin or India ink. These are some of the dye compounds which are very larger in size. They cannot penetrate inside the wall of the bacteria. As a result, the bacteria will not take any color. It is a negative staining, means the bacteria will not take color. Rather, the whole background will take the color of the stained compound. And then we can visualize the bacteria. So a little bit modification we can do it. What basically we do is, let us see it here. We just put a drop of nigrosin dye in a slide. And then we pick up the bacterial culture and mix up with the dye compound. And after that, using another glass slide, just like the blood smear, 45-degree angles, we can just make a smear. So once we prepare this particular smear, we can dry out this smear. Then very briefly, for 3 to 5 seconds, we can fix the bacteria with methanol. Okay, so after methanol fixation, to stain the interior of the bacteria, we can use this some dye like crystal violet. We can use it, and then we can dry it again and wash it in the tap water, and we can observe on the oil immersion objective. And these are the pictures what we can see it under the microscope. What will happen? The whole background will take color. The bacteria will not take color. And when you apply this crystal violet stain the second time, then what will happen? This inner size of the bacteria will take color, but the capsules will not take any color. So thereby we can visualize the capsulated bacteria by performing this negative staining. Let us see these slides another way. The background is taking the color, and inside the bacteria we can put another color, and the capsules will be very fractile. Okay, so all the compounds will be removed from the capsule dye compound. As a result, surrounding the bacteria, we can see a cleared area. This indicates these are the bacteria which are possessing capsule layer. Okay, so we know that the bacteria can be differentiated as capsulated and non-capsulated bacteria, and capsule is an influence factor of the bacteria, and they can initiate some disease. As I said, that certain bacterial capsules are too toxic for body cells and can cause massive RBC see disruptions because of this capsule layer. Many other things that there. Sometimes field we can see some capsulated and non-capsulated. I guess this the bacteria which are encapsulated. This is another non-capsulated. Brown bacteria are also non-capsulated, and this bacilli are capsulated. So for that purpose, we can perform this negative staining. Okay. But as I say that the negative staining on special purpose, like demonstration of capsules only, we go for this negative staining. Into simple staining, this is also not very frequently used. Only under the conditions like suppose suddenly if we just want to see the bacteria in the culture plate, like whether it is coccus or bacillus or what is this arrangement, without having an interest to understand more about the organism, then simply we can prepare a smear and stain the one dye compound, and then we can visualize it. So in simple staining, we use only one dye compound. So examples, lot many are there. 3 I have cited it here, like aqueous methylene blue, alkaline methylene blue, carbolfuchsin. These are the dye. There are many, like crystal violet, we can use it. Then gentian violet also, we can use it as a simple staining. So whatever the stain you will use it, the bacteria will take that color. Like if you use methylene blue, they will appear blue color. If you use carbolfuchsin, they will appear as red color. If you use saffronin, then they will take again red color. So in the simple staining, we can simply visualize the bacteria. Suppose here you can see this all bacteria, they are individually arranged and they are bacillus. So individual bacilli, you can see it here. So immediately we can understand the shape of the organism. That helps us in diagnosis purpose, okay? But it cannot provide more information about whether it is gram-positive or gram-negative, because this is a simple stain. Another example, you can see these are some bacteria which look like the coccobacilli, like the length and width is almost same, and they're appearing as coccobacilli. This is another structures which we explain it. And we know that our bacteria target or the bacteria of interest is coccobacilli, then yes, we can say that these are the coccobacilli. So here probably they use this dilute carbolfuchsin. So this is about the simple staining. How we do it? First, we prepare a smear, and previous class we learned about how to prepare a smear, all the specs. Then after drying, we need to fix it. How we fix it? We fix it by heat fixing, and what happened in the fixation, that also we discuss it. And then we apply the simple dye compound, only one dye compound at a time, and the bacteria will take that color, and after 1 to 2 minutes of staining, we wash it in tap water and then dry it, and we observe it under the microscopes, and we can see these organisms like this. Okay. Then the thirdly comes this differential staining. Here we give maximum emphasis because you must understand about this two staining technique. This is the most extensively used staining procedure in the microbiological laboratory. So one is called the Grams staining method, Grams method. And second is the Ziehl-Neelsen acid-fast staining method. Okay, Grams staining method is for all the type of bacteria. On the basis of this Grams staining method, Christian Gram in 1884, just remember, in 1884, he designed this staining technique. Can differentiate the bacteria into two broad categories as gram-positive and gram-negative. Okay, so this is the first primary things that we do in investigation of any bacterial infection to understand whether it is a gram-positive or gram-negative bacteria. Okay, so if we come to know that it's a gram-positive, then we give concentration on all the different gram-positive bacteria having similar characteristics likewise. So this the staining technique is a very simple 4-stage staining technique, which can differentiate the bacteria on the basis of its staining reaction, and we can designate them gram positive and gram negative, means which the organisms which actively take the primary color of gram staining method, we call them as the gram positive, whereas the bacteria which could not retain the primary color, rather they can take the counter stain, then we call them as the gram negative bacteria. So the biggest advantage of this kind of staining technique is that we can have a comprehensive idea about the organisms, like whether it is a coccus, whether it is a bacillus, whether it is spirillum, whether its arrangement is in chain or individual arrangement or in cluster, and at the same time whether it is a gram positive bacteria or a gram negative. So many information we can get it together with a very simple 4-stage staining method. That's about the gram staining method. Similarly, Ziehl Neelsen acid-fast staining is for acid-fast bacteria. Today only we'll be giving emphasis on the grams method. Let us just see. This gram staining is performed in a 4 very simple staining step. Okay, so if you are very carefully doing the staining techniques, and all the times you are going to get a very accurate results. So the primary stain that we use it in the gram staining technique is called as the crystal violet. Okay. This particular photograph, no doubt I have taken it from the internet, from Google search, but I had an objection on this particular pictures. Okay, my point is that when we use a crystal violet dye, the bacteria cannot take any purple color; it will take the violet color only. Okay, just think that this is the violet color. The first component of the gram staining technique is a dye compound called as crystal violet. So in the differential staining, we use 2 different dye compounds. Could you see here step 1 and step 4? This step 1, the first dye compound is called as the primary stain. This is called as the primary stain. And the second dye compound that we use it in the gram staining is called as the counter stain. Okay, primary stain and the counter stain. So in the gram staining technique, once we prepare the bacterial smear and fix it by applying heat, then we flood the smear with crystal violet and just keep it for 1 to 2 minutes. So within 1 to 2 minutes, what will happen? All the bacteria, irrespective of gram positive and gram negative, all the bacteria will take violet color. Okay, I had objections of purple. It cannot be purple, it should be violet. Okay, the bacteria, gram positive as well as gram negative, will take the same violet color. Okay, and the second component of gram staining is called as a mordant, M-O-R-D-A-N-T, mordant. And this mordant is a chemical mordant. Here we use it, is called as the grams iodine. It's an iodine solution, iodine and potassium iodide mixture. Now question arises, what is a mordant? Okay, if I just type it here, we call it a mordant. Okay, M-O-R-D-A-N-T, mordant. Now what is a mordant? So mordants are certain chemical or physical agent, certain chemical or physical agent which can help in fixation of primary stain. Again, I'm repeating the example of mordant. What is a mordant? Mordants are certain chemical or physical agent. Mind it, not all the time chemical. Here it is the chemical. Levels. It's a iodine solution that we use it. But in certain staining reactions, we use some physical agents like heat. Heat may also act as a mordant. So that's why we say mordants are certain chemicals or physical agent which helps in fixation of the primary stain. Helps in fixation, so that if you repeatedly wash this bacteria in running water, this bacteria will not lose the primary color. Although it is having the oxochrome group, but still, if you repeatedly wash it several times, they may lose all the color. So to fix this primary stain, we use a mordant. So the mordant that we use it in Grams staining technique is called as the Grams iodine solution. It is a mixture of iodine and potassium iodide. What basically this mordant iodine solutions will do, they will precipitate this crystal violet in the cytoplasm of the host cell. The dye compound will precipitate. Further, they will form some ionic compounds and they get precipitated so that they are not easily comes out of the bacterial wall when you wash it several times. Okay, so in actual staining technique, what we do, the smear, we flood it with crystal violet and then we wait it for 1 to 2 minutes. So after 1 to 2 minutes of incubation or staining, we discard the stain material and then we wash it in tap water briefly. After that, we apply this Grams iodine solution. So when you apply these iodine solutions, we apply it for hardly 30 seconds. 30 seconds is sufficient to precipitate the stain compound inside the bacterial cell wall. Yes. Pardon? You have some, what do you have? Okay. Okay, at what time it will start? Okay, all of you are going to join it. Okay, okay, then I should wind it up today. Is it? Okay, fine then. Then we'll continue our discussions in the next week class.
Thank you, sir.
Okay. So in a white background, you can see some colored organisms. So their shape, size, and arrangement, these things can be identified. That is the basic principle of putting color to the bacteria, staining for bacteria. At the same time, by the way of development and more improvement of the staining techniques, now we have a differential staining that we discussed in our last class about the different types of staining. The differential staining or selective staining, this is like most useful techniques for identifying and to study microorganisms, because differential staining, it will allow you to see the bacteria, to see its arrangement, but at the same time it can differentiate. That's why it's called differential staining. It can differentiate the bacteria on the basis of their cell wall component. And we know that broadly we have the two categories of bacteria, gram-positive and gram-negative. Okay, so those organisms, gram-positive bacteria, they will take the active primary stains of a differential staining, gram staining technique. That's why they call gram-positive, and the gram-negative, they cannot take it. And why it's so? It lies with the cell wall component. So in today's class, some of it we are going to discuss it, so that your concept on this differential staining become very clear. Okay, I just say that not only the differential staining, we had negative staining, one category, negative staining. This is, in fact, it's not an active staining, it's a negative. That's why it calls negative staining. Here the organism doesn't take any color, rather the background will take color, and we can see the unstained bacteria. That is another contrast, what we call as a negative contrast. So to mostly we perform it to visualize the capsules of capsulated bacteria. You know, the capsule is a very loose, gelatinous, amorphous material. They get deposited on the surface of the bacteria, and as such, they're so permeable to the color compound. If the color enters also, it will wash off in running tap water. So that concept, the negative staining, we perform it to visualize capsules of bacteria. Okay. Then simple staining, of course, if you're not going for or you're handling the organism and you know known organisms, so all the time no need to go for differential staining. Simple stainings are short duration, one-step staining process here. Just put one color, they will take the color, whatever the color you put it, and we can see this organism under microscope. Basically, to understand the arrangement and the shape, whether it is a coccus, whether it is bacilli, whether it is in chain, cluster. So these things we can identify by simple staining. But as you know, the simple staining cannot provide whether it is a gram positive or gram negative. For that, we have to go for differential staining, sometimes also called as selective staining. Okay. Then the last category of staining techniques that included in your syllabi is on spatial staining. So this spatial staining is basically meant for demonstration of certain spatial structure of the bacteria. More particularly, you know, there are certain bacteria which can transform into the most resistant form, what we call as endospore. So we are going to discuss about some of the highly fatal and pathogenic organisms under gram-positive category, which are producing the spores, endospores, and these endospores are their location, size, shapes help us in understanding and identifying those organisms. So spatial staining techniques include the endospore staining. Then again, you know, there are certain bacteria which possesses some cytoplasmic aggregates, what we call as the granules, velutin granules or metachromatic granules. So the presence of these spatial granules in their cytoplasm help us in identification of few selected bacteria. So for that, we can go for metachromatic staining and velutin staining so as to identify those bacteria. So basically, we can say these techniques: negative, simple, differential, spatial. So out of these, the first one is a negative staining. Bacteria doesn't take any color, whereas the rest, all the three, they take active staining. Okay. Can you hear me?
Yes, sir.
Okay, fine. Now coming to the first staining is called negative staining. So please note down this example of negative staining. Nigrosin and India ink, these are the two commonly used, either nigrosin or India ink, as a negative staining. So here what basically happened, these stain particles, the stain particles are so bigger that they cannot penetrate inside the bacteria. So if you make a suspension of the bacteria in nigrosin or India ink, the bacteria will not take any color, rather than the whole background will be colored or their color. So we can see in a contrast of negative contrast, like the background will be black and the organisms, you can see it under the microscope. So for these techniques, what we basically do, we just take a few drops of, again, here we don't prepare any bacterial smear that conventionally we prepared in the previous classes as we discussed, not like that. Here what we do in glass slide, we just put few drops of nigrosin or India ink. Then we pick up some bacterial culture, mix up with this stain, and then we make a smear just like how we prepare the blood smear. Okay. After that, we allow it to dry. Sometimes we can go for secondary staining of the bacterial cell wall with crystal violet. If not, also it doesn't matter. And after that, when you visualize this under microscopes, what you can see, the whole background is dark color, Negro or Indian color, and bacteria which is not taking any color will become as a refractile. So this is a way, what we call the negative staining. Rather than the bacteria, the background is colored, the bacteria doesn't have any, not taking any color. So this is the principle of negative staining. Here also you can see the secondary staining has been applied where the cell wall is taking the color, but we can see in between the cell wall and the periphery this non-stained area. This is the capsule possessed by this bacteria. So here you can see two bacteria together where the bacteria is taking the color and capsule is transparent. So this way, by producing our contrast, negative contrast, we can demonstrate the presence of the capsule. Let's see one more. Yeah, this is more clear. You can see these are all bacteria, and all the bacteria is surrounded with an empty space, this white area. This indicates this is the capsule layer. Okay, as I said, the capsule is highly porous. So if you wash it, if any stain particle enter, they will remove out of it. Okay, so here what they did is that first they have taken this negrosine stain, and then maybe I think this is they have taken what they might be Homasi stain, I guess. Yeah. In that, they make the bacterial suspension and they have prepared the smear, and after that they again put a primary simple stain so that the bacteria is taking the color, and the capsules after washing they wash off and they become clear. So this way negative stain is mainly performed to demonstrate the bacterial capsules. Please note it down. And this is why it's called negative stain, because the capsule doesn't take any stain here. Rather, this contrast is because of the background. Only here it is another slides where it can be demonstrated. The bacteria are taking some primary stain and the capsule is without any stain. So these are the capsule layer surrounding the bacteria. As you know, most of the capsulated bacteria are highly pathogenic. It is a virulence factor of the bacteria, but we cannot generalize. There are certain capsulated bacteria which doesn't they're not pathogenic here. Okay, in this particular slides, this is of course it is not an example of negative stain. Suddenly I just brought this slide here as because we are discussing about the demonstration of capsule in case the bacteria, and in fact this is a highly pathogenic bacteria that causes the disease anthrax in animal, as well as humans are also equally susceptible and equally fatal in human. Usually human infection is relatively rare, but if accidentally this bacteria from animal enter into the human, then it causes fatality. Okay, so these are Bacillus anthracis. They are also bacteria. We'll learn more about it. This bacteria possesses very important capsules, and this is capsules contain the virulence toxin, and because of which they can cause massive degeneration of the tissues, more particularly the blood cells, and can causes hemolysis, intravascular hemolysis. So to demonstrate these particular capsules, we perform a staining called as the Macfadien reaction. We'll learn more in details about the Macfadien reactions in the subsequent classes. It's something like when you use a polychrome methylene blue to stain the bacteria, then the bacterial capsules takes a pink color. So we can see surrounding the bacteria area. Usually, the bacterial capsules are made up of polysaccharide, but in case of Bacillus anthracis, it is containing some proteins, polypeptides are present. It's called as the polyglutamic acid, because of which it can take a light pink color when you stain with polychrome methylene blue. So this reaction is called Macfadien. Very simple staining reactions, but it's very important in diagnosis of the disease anthrax. Please note it down, Macfadien reaction. We'll talk more about it in the subsequent class when this anthrax disease diagnosis, we'll discuss it. At the same time, this particular bacteria is again can transform into the most resistant form, what we call as the endospores. In the nearby slides here, we can see these endospores, and then these are the endospores which are freely released out of the bacteria. When these are called as the free spores, but when the spores are remain within the shaft of the bacteria, for example, this particular area, if you see, this is sorry, it's a bacteria, and inside the exosporium, this endospores is present. We call it endospore, this one. And when this bacterial exosporium will dissolve and the spores will comes out of it, then we call it as a free spores, like this. This is a free spore. The spores are highly resistant for the bacteria. Resistant in the sense that the boiling water cannot kill it. Most of the disinfectant cannot destroy the spores. So for that, we have to go for a process what we call as autoclaving. We have to heat it up to 121 degrees centigrade and have to maintain for 15 minutes minimum of that. Only then they get destroyed. So these spores can survive in this environment for quite a long period. It's so amazing to know it that this Bacillus anthracis spore can survive in the environment up to 60 years. Probably it will be more. For last 60 years, this experiment is going on, and they found that the spores remain active till many years. And whenever they get access to the animal body, they will germinate to the vegetative state like this, and then they will just destroy the tissues and will kill the animal. So we'll come to this disease later part. Then coming to the simple staining. Simple staining is a staining technique, as I said, using only one kind of dye compound. Any kind of dye compound, but it should be only one dye. You just prepare a bacterial smear, fix it by applying gentle heat, and then you flood the smear with this any one of the stain, and just wait it for 1 to 2 minutes, and then wash it and see under the microscope. So whatever the color you will apply, the bacteria will take that color. So if you use a methylene blue, the bacteria will take blue color. If you use the carbolfuchsin, they will take the red color. If you use the safranin, the orange-red color, they will take likewise. So the purpose of performing simple staining is to just to visualize the bacteria, whether it is Coccus, Bacillus, or other morphology, or its arrangement. For example, here the bacteria, they are individually present. There is no any specific arrangement. All are bacilli. So these things we can study it by performing simple staining. Okay. So although here it seems to be a little bit longer, but these are not in chain. They are in the dividing stage. At a later part, they will get detached from each other, and then they will present scatteredly here. We had organisms what called as the Corynebacterium. Corynebacterium is an important group of bacteria are associated with varieties of disease condition in animals as well as in human. They produce some suppurative condition. So those Corynebacterium, they arrange just like the Chinese alphabet. So very commonly we call it the Chinese letter arrangement, just like the Chinese and Japanese script, the organisms they appear. So this is some way to describe And this helps us in identification of the organism. So let's see another one. Yeah, here in this, they have taken only one kind, probably carbofucsin they have taken, and all the bacteria is taking same color. And we can just see there very small bacilli. Sometimes the bacilli are so small that their length and widths are almost same. We call them as coccobacilli. Okay, so likewise, there are many. These are coccobacilli, where the length and widths are almost same, just to identify it. But this simple staining cannot provide you more information about the type of organism. Only the structurals you can study. Then comes the differential staining. As I said, the differential staining are the most widely used staining techniques in bacteriology, because it is a staining technique. It can visualize the bacteria. It can understand the shape of the organism. It can understand the arrangement of the organism. But at the same time, it can speak whether it is a gram-positive or a gram-negative bacteria. As you know, this is a first stage in disease diagnosis, bacterial disease diagnosis. We try to explore it whether the organism is a gram-positive or gram-negative. And then accordingly, we can proceed it for further confirmation of the organism. So Christian Gram in 1884, he developed this staining technique, and still we are using the same staining technique with some small modifications, and without which we are handicapped. We cannot go proceed and identify the organism. So Gram's staining technique is an example of differential staining, because it can differentiate gram-positive and gram-negative bacteria. So the main basis of this differentiation is the differences in their cell wall components. Okay. Similarly, we had another differential staining called as the Ziehl-Neelsen acid-fast staining method. Sorry. Yeah, Ziehl-Neelsen acid-fast staining method. This is specifically for the acid-fast bacteria. Then modified acid-fast staining. Okay. So this Ziehl-Neelsen, they modified the staining techniques for demonstration of acid-fast bacteria. As you know, there are few organisms which are all basically gram-positive bacteria, but their cell wall contains some specific chemicals, like mycolic acid is present in the cell wall of Mycobacterium that causes the tuberculosis disease. And this bacteria, it's very difficult to demonstrate by Gram's method. So another staining techniques has been identified, called as the Ziehl-Neelsen acid-fast staining method. So specifically, it can differentiate acid-fast and non-acid-fast bacteria. So it's an example of differential staining. And later on, some modifications of these staining techniques were developed, and we call them as the modified acid-fast staining, or shortly we call MZN staining, modified Ziehl-Neelsen staining, for demonstration of certain bacteria. So let's come to this first, the Gram staining. As I said, that Christian Gram in 1884, he developed this staining technique for differentiating the bacteria and categorize them as the gram-positive and gram-negative, and on the basis of their cellular component. Of course, at that time, he was not aware about why it is so. Simply by staining technique, he said that some of the bacteria are taking the primary color. He call them as the gram-positive bacteria, whereas there are certain bacteria which are taking only the color of counterstain, safranin, and he started calling them as the gram-negative bacteria. Later on, it was explored that this is because of the differences in the cell wall components of gram-positive and gram-negative bacteria. So as such, in this staining technique, we had 4 components. The first one we call as the primary stain, and the primary stain that we use in the ground staining technique is crystal violet. Okay, of course there are many modifications and some formulation has been developed, but basically all over the world people use these are the components in ground staining technique. Crystal violet is the primary stain. Primary stain is given the beginning of the staining procedure. Okay. So the second component of the ground staining is called as mordants. What do you mean by mordants? These mordants are certain chemical or physical agent which helps in fixation of the primary stain. We call them as mordant. Let me repeat it. The definition of mordant is mordants are certain physical or chemical agent which fixes the primary stain. So if you use the crystal violet as primary stain and then we apply a mordant, in case of ground staining technique, the mordant is a chemical agent. It is the Gram's iodine solution. It's a mixture of iodine and potassium iodide. And this particular formulation, iodine and potassium iodides, are earlier extensively used in veterinary practice as an antiseptic agents, as well as it can be given intravenously when for promoting the growth of animal, and even for dressing and flushing of uterus after delivery with this iodine and potassium iodide solution. So a modification of it, what we call as the Gram's iodine solution, act as a mordant. It helps in fixation of primary stain. Now let me tell you what will happen if you do not use a mordant. So if you just go for staining the slide with primary stain and then with crystal violet, and then if you wash it on the slides for prolonged period, then what will happen? All the primary stains will wash off. But when you use a mordant, then this primary stain will retain inside the bacteria. It will help in fixation of the primary stain. Okay, so the second component of ground staining technique is mordant. Okay, now coming to the third component of ground staining technique. This is a very crucial component, and here the differences occur. Okay, so it's called the decolorizer. So after applying the mordants, what we do, we flood the smear with decolorizing agent, and the decolorizer that we use most commonly in the ground staining technique is alcohol solution. So basically 70% ethanol we use it. If not, then we can go either or. If alcohol, or then we can go for a mixture of acetone and alcohol, 1:1 percentage. Okay. So this percentage and concentrations are important because it again related to how long you will decolorize this slide. So if you go for prolonged decolorizations, then even you can get some false negative results. Okay. So in case of the ground staining technique, the primary stain we use a crystal violet, and we the smear we just apply the primary stain crystal violet, and for 1 to 2 minutes we allow it to put the color. Okay, so within these 2 minutes, all the bacteria will take the color of crystal violet. Okay, after that we discard the crystal violet stain briefly. We can wash it in tap water, and then we can apply Gram's iodine solution. Okay, this Gram's iodine solutions we apply hardly for say 30 seconds. It's sufficient. Half minute is sufficient. 30 seconds we use it. So within these 30 seconds, these primary stains get precipitated inside the bacterial structures, and that helps in fixation. After that, we use the decolorizer. It's alcohol solution. So if you use 70% ethanol, maximum we can go up to 30 seconds, not beyond that. In some laboratory, people practice for a stronger concentration of alcohol. Say sometimes they use it even 95% ethanol. So if you use 95% ethanol, then the destaining period is only for 10 seconds, not more than that. So what I mean is that this concentration of alcohol depends on how long you are going for decolorations of the slides. Okay, so if you prolong the decoloration with 95% ethanol more than 10 seconds, then what will happen? Even the gram-positive organism also start losing the color. So this is a very crucial stage in the gram staining, the decoloration stage. Okay, even if you use acetone alcohol mixture 1:1, we shouldn't go more than 10-second time for decoloration. Okay, so once the decoloration is over, we wash it briefly and then we apply counterstain safranin. So why this is the last component we call the counterstain. What basically happened? Let us see in the diagrammatic in this particular picture, so slide. When you applied the primary stain crystal violet, all the bacteria, irrespective of gram positive, gram negative, they will take the color of crystal violet. This is my objection. It should not be purple, it should be violet because you're using crystal violet. So they will take the violet color. And then you apply the iodine for 30 seconds. So it will help in fixing the primary stain. Then it comes to decolorization. This is the most crucial stage where the differences occur. Okay, so when we apply this alcohol, either 70% ethanol or 1:1 acetone alcohol, or even 95% ethanol, when you use it for a specific time period, the gram-positive bacteria will not lose the color. Why? It is because of their thick cell wall. Whereas the gram-negative bacteria, they will immediately lose the color. The alcohol will penetrate and they will wash off all the stain. Although it has been fixed by iodine, but still the stain will, the bacteria will lose this color because of this alcohol effect. The gram-positive, they doesn't, here the difference comes. That's why we call that the primary stain is actively taken by the bacteria. We call gram-positive, and here they lose it. That's why we call as a gram-negative. Okay, so once this bacteria, gram-negative bacteria, initially took the color and decoloration, they lose the color. Then when you apply the safranin, immediately they will pick up the safranin color. Safranin is a red color. Crystal violet is violet in color. So since the gram-positive bacteria already they have this color, that's why they are no more taking the safranin color. As a result, at the end of the gram staining, we can see the gram-positive bacteria as violet color, whereas the gram-negative bacteria will appear as a red color, orange red color. So looking at the color, we can say that this is a gram-positive and this is the gram-negative. So if you go theoretically, you have learned about these things. This is a schematic diagram. The cell wall, as I said, that the main principle lies with the cell wall component. The differences of the cell wall component of gram-positive and gram-negative. So if you recollect it, then you'll find it that in case of the gram-positive bacteria, they have a very thick peptidoglycan layer. So as you know, these peptidoglycans are formed by N-acetyl glucosamine and N-acetyl muramic acid component. They are linked by beta 1,4 glycosidic linkase, and then this muramic acid component, they have this peptide site chains through which they cross-link each other. If you see, these are the cross-linking you can see. So in case of the gram-positive bacteria, there are many strands of peptidoglycan. They polymerize to form a thick peptidoglycan layer. Okay, and there are more cross-linking within this layer. As a result, in case of gram-positive bacteria, the cell wall is very tight. So when you decolorize it, alcohol cannot penetrate deep inside the bacteria and cannot remove the color. because of this thick peptidoglycan and more cross-linking of the peptidoglycan. Whereas if you just comparatively see the gram-negative bacterial cell wall, comparatively the peptidoglycan layer is very thin. This is the part, only few strands of peptidoglycan polymer you'll get it, and there is less cross-linking among these strands, so peptidoglycan is thin. However, the gram-negative bacteria, they possess the additional outer membrane, which is absent in the gram-positive bacteria. Although they possess an outer membrane outside of this peptidoglycan, but this outer membrane is basically composed of lipids and phospholipids. So heavy amount of lipids is present in the outer membrane. As you know, the lipids are easily dissolved by alcohol. So when you apply, in case of the gram-negative bacteria, this alcohol readily dissolve this lipid layer, and then they will percolate through this peptidoglycan, which is a loose structure in case of gram-negative, and they will decolorize the entire bacteria. So thereby what happened? The gram-negative bacteria will lose the color and gram-positive will retain. So once they will lose the color, when the next stage you will apply the safranin, so they will immediately pick up the saffron color. It's a very simple staining techniques, but so useful that we can identify and you can see the gram-positive and gram-negative bacteria in the microscopic field. So this is another picture showing these cell components. Here another schematic that at the beginning, when you prepare the bacterial smear, bacteria doesn't have any color on stained refractile organism. Then you apply color, crystal violet, both gram-positive, gram-negative will take the color. Then you apply iodine, and then when you go for decolorization, gram-negative will lose the color because of their thin peptidoglycan layer. And when you apply the counter stain, the gram-negative bacteria will take the color of counter stain, not the gram-positive bacteria, which already taken the primary color. So likewise, we can differentiate. And here it's a picture you can see when you prepare the smear. These are the bacteria gram-positive, which is taking this violet color, and these are the gram-negative bacteria, which is taking the saffron color. So looking at this color, immediately you can say this particular microscopic field contain both gram-positive and gram-negative bacteria. Okay, and all of them, they are basically bacilli, no coccus is seen. So thereby we can differentiate it. So this is in principle just taken from your practical manual. You can go through it. I think all of you can understand it now. Then coming to the Neelsen acid fast staining. So this is a difficulty appeared when the mycobacterium species that causes the tuberculosis in man and animal. This bacteria, when the people tried to visualize it and stain it with the gram staining, they found that the bacteria is not taking the gram stain properly. After performing the gram staining, some part of the bacteria take color, and majority of them, they are refractile, doesn't take any color. So in that process, this particular staining techniques was developed, and the principle lies behind this staining technique is that the bacteria which possesses mycolic acid, they polymerize with certain cell wall lipid, complex type of lipid, and they made the bacteria impermeable to most of the common stain, like gram staining. Okay, so it has been seen that if you apply heat and then you use a very strong color, like concentrated carbolfuchsin, it's contain the carbolic acid, very strong stain. It imparts a very intense red color. Then the bacteria can be stained, and once the bacteria pick up the stain, it is extremely difficult to decolorize it. Once they take the color, it is difficult to decolorize it. That becomes the basis of acid-fast staining. So why we call as the acid-fast staining? Because very strong decolorizing agents like acid alcohol. You know the alcohol is a good decolorizing agent, but if you mix up acid and alcohol, that becomes a very, very strong decolorizing agent. So once the bacteria is stained with strong carbofuchsin, then even the most strong decolorizing agent cannot decolorize the bacteria, and the mycobacteria containing the mycolic acid will not get decolorized, whereas all other bacteria will readily lose the color, and that becomes the differential stage in case of the acid-fast staining technique. And this is the principle of acid-fast staining. Now let us see what way it is different, the mycobacteria. It's quite interesting. This mycobacteria, although they're cytochemically gram-positive bacteria, but they contain heavy amount of lipids on their cell wall. This is a basic things or differentiating point in case of gram-positive and gram-negative bacteria. The gram-positive bacteria, they possesses very less amount of lipid, whereas in case of the gram-negative, they contain heavy amount of lipid, but with some exception. Exception is this Mycobacterium species. Mycobacterium, cytochemically, it is a gram-positive bacteria. They have a thick peptidoglycan and more cross-linking, but they contain heavy amount of lipid. This is an exceptional thing, like arabinogalactan, which polymerize with the mycolic acid, and they give rise to a component called as the WOX-D component. It is a waxy substance, wax-like substance. As you know, the wax, when you apply heat, then this wax melts, and then when it cools down, then it gets solidified. So this arabinogalactan along with mycolic acid, they polymerize to form a compound called as the WOX-D, and which makes the bacteria impermeable to the regular stain. Okay, so what are different components of the acid-fast staining? Just like the gram staining, you'll get two different color. All the your differential staining, we use more than one staining component stain per color. Here, like carbofuchsin, and we use a contrasting methylene blue. Okay. In case of the ground staining, if you can recollect, we use the crystal violet and saffronin. Mind it, we need a contrasting color. Suppose carbofuchsin gives us the intense red color, methylene blue gives us the blue color. In case of the ground staining, crystal violet gives the violet color, and the saffronin gives us the red color. So this color should be contrasting so that we can differentiate the color under the microscopes after the staining. So the primary stain that we use it in the acid-fast staining is concentrated carbofuchsin, or sometimes we call it the strong carbofuchsin. This carbofuchsins, we prepare the bacterial smear. Then we flood this bacterial smear with the carbofuchsin. Then here it is very interesting. We apply heat. This heat acts as a mordant. Please note it down. When I say the definition of mordant, I say that the mordants are certain physical or chemical agent that helps in fixation of primary stain. So in case of the ground staining technique, the mordant was a chemical agent, but here in acid-fast staining, it is a physical agent. It is heat. It says that when you apply heat, then this WOX-D components become permeable. The mycolic acid layer become permeable to the carbofuchsin stain, and the stain penetrates and they impart color to the entire bacteria when you apply heat. So it becomes permanent staining for the bacteria. So this heat simply acts as a physical agent in the fixation of primary stain in acid-fast staining. So after heating, heat application, say carbofuchsins, we flood the smear with carbofuchsin, then we apply heat, and this heat should be a gentle heat. So in the subsequent two videos, we'll see how the heat can be applied. So if you heat it up, the stain and the smear for say 3 to 5 minutes, so what will happen? The bacteria will take the color of carbofuchsin, and all bacteria will take the color. Then we can go for the decolorization stage, and the decolorizer is a very strong decolorizing agent. We use the acid alcohol. Okay, so as I said, the alcohol itself acts as a decolorizing agent in the acid ground staining, but here we use a mixture of 97% ethanol and 3% hydrochloric acid or sulfuric acid. So any one we can take it. There are certain procedures where instead of acid alcohol, they simply go for acid solution, 20% sulfuric acid solutions. They use it for decolorizations. There are some modifications, and laboratory-wise, these variations are there. And so, because we are using an acid solutions for decolorization, that's why this staining technique is called as the acid-fast staining. Okay. The meaning of fast is that the bacteria can prevent decolorization by acid solution. That is the meaning of acid-fast staining. Fast means prevent decolorization by acid solution, strong acid solution. That's the main basis of acid-fast staining procedure. Okay. So here again, since the primary stain, once the bacteria will take the color of carbofuchsin, it is extremely difficult to remove the color. So there is no any definite timing of decolorizer, application of decolorizer. Unlike in ground staining, we say that the timing is very, very crucial. Like if you use 70%, we can go up to 30 seconds. If you go for higher concentration or acetone alcohol, then we should go for 10 seconds, not beyond that. And that will affect the staining properties of the bacteria in case of the ground staining. But here, in case of acid-fast staining, this timing doesn't matter, because this carbofuchsin staining itself is a very permanent kind, and then we go for applying this decolorizer till the whole smear become clear. We go on applying this decolorizer. Usually say 1 to 2 minutes is sufficient for decolorizing. Then we wash the slides, and then we apply the counter stain. As you know, all the non-acid fast bacteria, although they will take the color at the beginning stage, but when you apply this acid alcohol, non-acid fast bacteria will readily decolorize. The color will be removed. But because of presence of mycolic acids and arabinogalactan forming the wax stick component on the cell wall, the acid fast bacteria will not decolorize. Here the difference comes. So finally, we can use a contrasting blue color. So low fluorescent methylene blue, we use it. And then all the non-acid fast bacteria will take the methylene blue color, and the acid fast bacteria will still retaining the color of carbofuchsin. So thereby, we can identify very pathogenic bacteria having tremendous zoonotic importance, and the tuberculosis disease can be diagnosed. And it becomes so much popular and very easy to perform it, and almost in all diagnostic laboratory, they go for the simple 4-stage staining techniques for identification of mycobacteria. Let's see video lecture. I guess all of you can hear me.
Yes, sir.
Okay. Yeah. The acid-fast staining techniques. Of course, this videos I have just taken it from YouTube. So some points I'm not happy with this video, but still I can explain it, the things. So we need a flame for heating up, but here the heating procedure is different. They're applying direct heat by passing through the glass slide, but it's not a preferred method. And moreover, in this staining techniques, you can see the persons are doing the staining without wearing any gloves. It's not recommended. One is we need a protection to the eyes by applying Google spectacles, and then we should use some gloves so as to protect our hand, because this strong carbon fuchsides can immediately impart color to the our body cells. So using 20% sulfuric acid is also risky affairs, so have to be careful. Here what they are doing is that they are preparing a bacterial smear, and they are taking this 3 components. These are the 3 things they needed. This is the smear they prepared. After preparing the smear, they have fixed it by applying heat, and then in the staining rack they just flood it with the stain, and then they are applying heat by passing this spirit lamp just below the slides. So this is a risky affair, and again, without wearing any gloves they are handling it. It's not recommended. So here is showing how we can apply the heat, but I will go for another video where the appropriate methods I will show you. Only things you need to take care is that this stain should not boil. No bubbling should be there when you apply heat. Okay, overheat, or if it get boils, then the smears may comes out and you cannot see the bacteria properly. So I mean, you pass the spirit lamp 3-4 times and just wait and again pass it. Likewise, we can heat it up to 3 minutes, and then the bacteria will take the color. By applying this heat during this particular stage, what will happen? The mycolic acid or the wax stick component become permeable, and the stain will penetrate inside the bacteria, mycobacteria. After that, we can, as showing the second time, it's applying. But this is a risky affair. Instead, the steam boil water, steam vapor is better than that. We'll see in the next video. Then we can wash the slides with running tap water. So by this 3 to 5 minutes of staining, the bacteria will take the color. So this is a washing stage as they're washing the slides. So after completing the washing, we can apply Decolorizer. This is the stain, the smear. Now we can apply the Decolorizer. This Decolorizer here, they're using the 20% sulfuric acid. It's a very strong decolorizing acid solution. So after decolorations, we can wash it in the tap water, and then here the timing is not much crucial, because once the bacteria will take the color, they will never get decolorized by acid alcohol acid fast bacteria, but all other non-acid fast bacteria will readily become decolorized. And then the counter stain is applied here, the Loeffler's alkaline methylene blue. So yeah, minimum of 1 minute we should keep it, and then we can wash it thoroughly, and we can see the smear under the microscope. So these are the simple stages, and then we dry this smear by blotting paper, and it is now ready for visualization under microscopes. So here it is another this method I personally prefer, and we'll be doing in our practical class. While doing it, I just took it from the YouTube's, so showing it how to wear the goggles just as a protective wear, because these solutions are very, very caustic, and then they may cause damage if spill over to face or hand. So Again, the timing another varies from lab to lab, and there is in crystal violet instead of low fluorescence alkaline methylene blue, but we'll learn about this low fluorescence alkaline methylene blue only. Yeah. Yeah, this is the technique. What I want to show you that we can just boil water in a trough, and these are the smears we prepared it. This is a boiling water trough, and steam is arising continuously. So this steam heat is the safest and the best way for applying heat. There is no chance of boiling the carbon fuchsines in this particular method. So she's just covering it with blotting paper, and then she'll be applying strong carbon fuchsine above the smear. So boiling water trough is a better, steam heat is a better in this staining technique. So she's using the carbon fuchsine and she's applying above it with a dropper, wearing gloves properly. This is the right way. Then applying 3 to 5 minutes. And now it is time to wash it. So after washing, the running tap water, we can apply decolorizer. And when it gets decolorized, so likewise is applying decolorizer. So all non-acid fast bacteria will decolorize readily, whereas the acid fast bacteria, they will still retain the color of carbon-fuchsine. Okay. Then we can just use this. She's using crystal violet, but it's a bit low fluorescence alkaline methylene blue as per our protocol. And again, washing, and the slides is now ready for visualize under the microscope. This is another video. This two video is skip it. So once it over, then we can see it. And these are some smear from maybe from sputum. Like, you know, in case of the human, the tuberculosis nodule, when they form in the lung, it causes constant irritations, and that leads to coughing, and sometimes this nodular lesion get burst inside the lung, and the blood comes out through mouth during coughing. So the saliva also carry a large number of this organism, and if you can demonstrate the acid fast bacteria like this way, these are the slide, which is diagnostically confirmatory in nature. So the acid fast bacteria will take this carbon fuchsine color, okay, whereas the non-acid fast bacteria will take the blue color. So likewise, by performing this simple staining technique here, you can see the filamentous form of the acid fast bacteria. They are getting aggregated in the impression smear, taking it from certain organs or some clinical specimen. You can see these are the cord, like this mycobacteria we learned in the theory when we discuss about the disease tuberculosis. They form some cord-like structures, and just like ponces, you can see it in the microscopic field. The acid fast. This is a full of acid fast bacteria. This might be a full-grown tuberculosis cases in. So many number of bacteria, we can see it in this microscopic field. So thereby it help us in differentiation and identification of specific bacteria. This is Mycobacterium leprae that causes the disease leprosy in human. So comparative structural differences of this gram-positive, gram-negative, and Mycobacteria, they try to show it here. About this peptidoglycan and other components. So all the staining reactions are basically depends on the cell wall structures of the bacteria, and that helps us in getting the differential staining. Then comes to the special staining. Special staining performed just to demonstrate the special structure of the body, just as endospores in case of the spore-forming bacteria, as I said, the metachromatic granule staining, and in some cases the flagellar staining. Of course, it's not very successful, but still, in case of certain bacterial flagella, we can demonstrate it with a special staining. So here we'll be mainly discussing about the endospore staining, and we can see it and perform this staining. So there are basically two techniques we use it. One is the modified Tildenson technique. It's a modification of acid fast staining. Here also with the same principles, like it is very difficult to stain the bacterial spores, but once they will pick up the color, then it is very difficult to decolorize them. This is the basic principle of spore staining. So using this Ziehl-Neelsen carbofuchsins, we can stain the bacterial spores, and later on we can apply the decolorizer, and in counter stain we can stain the exosporia. So it's a modified Ziehl-Neelsen technique. Similarly, we had another, is called as the Schaefer-Fulton method, where we use the malachite green instead of carbofuchsin. We can use the malachite green stain for staining the spores. So it's very difficult to put color to the spore, but once they will pick up the color, it is extremely difficult to decolorize them. That is the basic principle. I guess all of you understand about the bacterial spores, how it is formed. Not whole bacteria, mind it. It's a property of only few bacteria which can undergo endospore formations, and this is the most resistant part, and they contain heavy amount of dipicolinic acid on the surrounding of the genetic materials, and calcium dipicolinic acid get deposited. That gives us a very thick crust surrounding the bacteria, and they remain in the dormant state. Okay, the spores are metabolically dormant. There's no metabolic activity goes on inside, so simply they undergo a dormancy, and this dormancy for certain bacteria may last for many years. That's a most worried point. And then once they get the favorable condition, the endospores can germinate into a vegetative form and can produce disease. So in this modified Ziehl-Neelsen method, we use the strong carbofuchsin for staining the bacterial smear, and we go for a weaker decolorizer. 0.5% sulfuric acids are used. This is the modification. That's why it's called the modified Ziehl-Neelsen method, and we can use the method below. This is a procedure, simple 3-stage process. Of course, this pictures, I think this is for malachite green staining only, the Schaefer-Fulton method. So we can flood the smear with malachite green, and we can apply heat, steam heat. So all the bacteria will take, including the spores, will take this color of malachite green. Then when you go for decolorization, the spores will not lose the color. This is the differential stage. But all other vegetative form of bacteria, they will get decolorized. And when you apply this counter stain, say safranin or any color, because it is green, contrast will be the red color. Again, the bacteria exosporium will take the color. So within the bacteria, we can see the spores. The location of the spores within the bacteria are important to identify the organisms. As you know, you have probably heard about the drumstick bacilli. The disease that all of you know, we call it as a tetanus, Clostridium tetani. It causes a highly fatal disease in man and animal. We call it a tetanus. And mainly through accidental injury, the bacteria get entry to the body, and then they can produce toxins, and the toxins effect is fatal. They may leads to death of man and animal. So that particular Clostridium tetani, they possess terminal. The spores are present at the terminus of the bacteria, not like this. Even it's at the end, one end, and they are bulging type, and it's a round, look like a drumstick. Beating drumstick, like a stick with a bulb at one end. We call them as a drumstick bacilli. So that special structures help us in identification of the bacteria in the clinical specimen. Whereas we have certain spore-forming, endospore-forming bacteria like Clostridium chove that causes the disease black quarter in large animals, and this is some sort of necrotic myositis occur. Whole muscles in the leg and the thigh become rotten from inside because of that bacterial infection, Clostridium chove. So that produces a central spore. In the spore present at the center of the bacteria, and it is almond shaped, oval shaped. So these criteria are helpful in identification of bacteria. So for that, to see whether it is central, subterminal, or terminal, it's a bulging, non-bulging, it's oval, round. So this characteristic, to study these characteristics of the organism, we go for the spore staining. So likewise, different type of stainings are performed to visualize and diagnosis of bacterial diseases. So in today's class, I prepared up this part. In the next class, we'll go for the other part like sterilization, disinfections, and preparation of media, etc. Okay, so up to this point only, we'll consider the staining class. Thank you very much for attending the class. If you have any questions, we can have some interaction. 34 students only joined, very less amount. I don't know why. Do you have any specific question? You can switch on your microphone and can ask question. Yeah, if not, you can just send these questions in my email address, or you can send it by WhatsApp, so that in the next class we can address these questions and have more discussion. Thank you very much. I'm winding up today's class. Thank you.
Thank you, sir. Thank you, sir.
Thank you, sir.